Good morning, Biology 102. Today we are talking about the four macromolecules that build all living things. The four macromolecules, you actually know them because you have to eat many of them every day in order to live. So they're on these nutrition labels that I show in this particular diagram. The four macromolecules are carbohydrates, lipids, which is a fancy word for fats, proteins, and nucleic acids. You might know nucleic acids as DNA and RNA, the actual polymer type. I want to go through first the similarities between all four macromolecules, what they have in common. So all four macromolecules often share a carbon-based backbone in some piece of their chemical. We say that carbon is the backbone of life, and it has to do with the carbon being the backbone of these macromolecules. And these macromolecules, you can imagine, are kind of like Lego bricks that we can put together in various ways to build a bacteria or a fungus or a tree or a human, right? So all different kinds of living things. All living things are going to use these four macromolecules in order to build their structures and life, right? These carbon-based backbones can bond to other things like hydrogen and oxygen, other carbon, nitrogen. And so remember, 96% of all living matter has that shown, those carbon, hydrogen, oxygen, nitrogen elements in it. And so it's quite nice that we can bond and make all different patterns with these very commonly found elements around Earth's surface. All four macromolecules have simple repeating units called monomers that then link together into more complicated structures called polymers. Those structures can be sheets and chains. Think about like your body, things like helices and hair and skin, these sheets and fingernails. Those are all different kinds of polymer structures that build up like who you are. Over here, I show this pearl necklace with green pearls on it. So you can imagine each of these pearls being a monomer, and then we're linking them together into more complicated structures called polymers. Now, all four macromolecules are built into polymers using a process called dehydration. And dehydration is naming what it is doing. Dehydration means removing water, right? Or losing water. So these macromolecules will have an OH group on one end and H group on the other. If we combine those and remove those, we can remove water and then link one of these green pearls to a larger structure. So this takes shorter monomer pieces and links them into larger polymer structures. So all four macromolecules is going to use this process of dehydration in one form or another in order to build these more complicated structures. And then we also break down the four macromolecules using a process called hydrolysis. And hydrolysis, again, is naming what it is doing. Hydro means water, lysis means to break. So in this particular reaction, we are actually adding water across a bond. We're going to add an OH and an H group, and then we're going to break a polymer, a big, large structure, into simpler monomer pieces or simpler pieces. So all four macromolecules use these reactions to either build them into polymers or break them down into monomers. On your human body, you can imagine like when you add different like structures like hair and skin and things like that, you are doing dehydration reactions to sort of create those polymers at your surface. Hydrolysis would be happening inside of your gut, breaking down all of those kinds of things like hamburgers into smaller pieces so that you can then use those smaller pieces to build you. You can't just slap a hamburger on your hand and grow, right? You have to break that hamburger down and then use those pieces to rebuild as a human. And so hydrolysis allows for the gut to break down those foods that we are eating. Now I want to go through all four macromolecules and share with you their monomer structure, the polymer structure, and a function for each of those macromolecules. We're going to start with carbohydrates, and on your study sheets there's actually a table that you can fill out with each of these pieces: monomers, polymers, and function. And I want you to know those particular pieces for each. So carbohydrates are made up of a monomer structure called a monosaccharide. That is a single unit sugar. Some common monomers that you might have heard of are glucose, fructose, and galactose. I show glucose over here, and you can see it has a linear form but also a ring form that is quite common. We can then link these monosaccharides into simple sugars called disaccharides. So that's like two of them linked together, like sucrose or table sugar is glucose and fructose. Those two sugars bonded together. Over here I'm showing lactose, which is glucose and galactose, as a disaccharide. Or you can link sugars into hundreds to thousands of units, glucoses or whatnot, long, and come up with complex carbohydrates like your starches. That's a common storage form for plants. Glycogen is how animals often store their complex carbohydrates in our livers and things like that. And then cellulose builds like cell walls of plants. We call it fiber in our diet, but that's also a complex carbohydrates that plants use in their particular lives. Lastly, the function of carbohydrates. We know carbohydrates are often called our quick energy source. They're often our go-to energy. It's the place that our cells are going to go to first. We're going to break down those simple sugars in order to form our energy that is necessary for our life work, right? Those complex carbohydrates can be used. Cellulose, again, is used for plant cell walls, so an outside cellular structure. Fungal cell walls and exoskeletons, the outside surfaces of insects, are built from another complex carbohydrate called a chitin. And then sometimes carbohydrates can serve as signals inside of our body, telling other cells either what they are, that they belong, that they're self, so they belong to you, or signal other things for like sort of cell-to-cell communication. A good example of a cell signal you might have heard of is ABO blood type. If you have A blood type, you have a certain structure of a sugar on the outside that we say is A. If you have B blood type, you have another shape of a sugar on the outside of your red blood cell. And if you're O, you're lacking a sugar. And those sugars then for red blood cells serve as signals for your immune system cells and other cells in your blood. Next is lipids. This is a fancy name for fats, and the monomer structures, it kind of depends on the class of lipid that you're talking about. So this is kind of a diverse group, and there are many different kinds of classes of lipids. But one group is built from a triglyceride. Over here I show a glycerol head with 3 fatty acid tails. That's known as a triglyceride. And another sort of group comes from kind of a cholesterol-based chemical with 4 rings. And so that also builds a few sort of polymer structures. So from a triglyceride, that monomer structure, we can build something called a saturated or an unsaturated fat. If we look back at this particular picture of a triglyceride, if you have maximum number of hydrogen on this outside sort of carbon base unit, then you are saturated with hydrogen, and we call you a saturated fat. If you are missing some of these hydrogen, then you are called unsaturated. And every time you're missing a hydrogen, it actually gives a double bond between those carbons, and it actually kinks the tail. And so you get these kinky tails. Saturated fats are kind of a linear structure. They get a bad rap with like cardiovascular surgeons because they can stack, because they're so linear, they can stack so nice and neat in our arteries. Unsaturated fats have these kinky tails. They don't stack as nice and neat. They are often liquids at room temperature. They're things like olive oil, some of those oils. And so they are the preferred, it's what your heart doctor is going to tell you to like eat because they don't stack so nice and neat. Cholesterol, I'm showing in this particular picture up here, you will notice that it has this 4-ring central area, but if we change these external structures, we actually get different kinds of hormones and steroids, like this is showing estrogen over here, or testosterone. And so cholesterol is something that is used for cellular structure, but also can be used and like kind of changed and altered ever so slightly to build different kinds of steroids and hormones that we see that are commonly used in and around the human body, but in and around all living things. Lastly, the functions of lipid. If carbohydrates are your quick energy sources, usually we say that lipids are your stored or long-term energy. So these are fats and oils that we keep around so that we have energy on tap in case we run out of our carbohydrates. Another big function for lipids is they build these cell membranes. They can also be components in insulation, so blubbers and things like that, to keep our heat consistent, waxes like in our ears, things like that help us. And they can also serve as cell signals, so be some kind of signaling system from cell to cell communication. Third macromolecule is a protein, and the monomers are called amino acids. There are 20 common amino acids that make up all different living things, plus there's a few rare ones as well. An amino acid has an amine group, this nitrogen group on it, as well as this carboxyl group, and then they are named for their side chains. A lot of amino acids you've probably heard of, like phenylalanine, methionine, something like tryptophan. So you might have heard of those particular sort of amino acids. If we group amino acids together into chains and sheets and circles, all kinds of shapes, then we get something called a polypeptide or a protein. Over here, I'm showing you how this dehydration reaction works. If we pull an OH group from the carboxyl, the COOH group, and an H group from the amine group, then water comes off, and in its place, we can link the carbon to the nitrogen in a peptide bond. And this is how we build our proteins into sort of more complicated structures. Lastly, the functions of proteins. Proteins are really important for the human body. They build primarily these chemicals called enzymes that speed up and facilitate our chemical reactions. So we have lots of different enzymes that run our bodies, right? Proteins are also structurally important, like muscles and skin and fingernails, hair. All of these structures are, they often will have a protein component to them. So a lot of like what you're seeing as your protein traits on the surface and structurally holding like sort of a living thing together will be in part proteins. Proteins help us store amino acids. They are also, although the membrane is mostly a lipid, if we want doorways or gates into cells, then those are often proteins. So transport gates and things like that on the cellular surface are also made of protein. And lastly, cell signals. So just like the carbohydrates A, B, O blood type is a sugar on your red blood cell, you also have the positive, negative, like your A positive or A negative blood type. And that negative is a group of factors that are proteins that are also found on the red blood cell that signal to other cells. And so proteins can also be a part of that communication. The last kind of macromolecule is a nucleic acid. The monomer is called a nucleotide. And you probably know nucleic acids by their polymer forms. So DNA, you've probably heard of, or RNA. So the repeating unit in DNA and RNA is called a nucleotide. Nucleotides have three pieces. They have a sugar piece here in blue, which looks kind of like a house. They have a phosphate group in yellow, and then they have a nitrogen base that comes in a variety of forms. It's almost like an L-shaped chemical, right? With the sugar in the middle, the phosphate, and the sort of nitrogen base. So these nucleotides contain these three chemicals. When we join them together, we can make DNA, which is also known as deoxyribonucleic acid. It has deoxyribose as its sugar, so that's where we get its name. Its nitrogen bases are adenine, thymine, cytosine, and guanine, so A, T, C's, and G's. And we say that it is a double helix because these letter L's, they repeat sort of in a double, almost ladder. So you have two sides of them in this like kind of ladder structure to form DNA. RNA is ribonucleic acid, the sugar is ribose. We switch out one of the nitrogen bases, thymine, for uracil, and it is a single side. So imagine if you cut a ladder in half and you have that single-sided structure, then that's what you're looking for. When we look at the functions for these two kinds of nucleic acids, DNA primarily is like the blueprint, the directions. It houses your genetic code that's passed from parents to offspring, and it's the directions to build you with all of your protein traits. RNA, on the other hand, is almost like DNA's minion, right? It's a worker bee. There's a variety of kinds of RNA, and then they help to build your protein traits. So DNA is like the instructions for building those protein traits, and then RNA is the worker bee or the minion. DNA could tell it, hey, build me some insulin today, right? And then RNA will be the kind of nucleic acid that then within your cell builds that insulin so that you have it to regulate your blood sugar. Those are the 4 macromolecules. Again, I want you to know the monomer, the polymer structures, and then also a function or a group of functions for each of those. There is a table on your sheet that you can complete. Otherwise, have a great day, and I'll see you next time.